Thanks for the introduction. So I'm currently a postdoc at the Electronic Visions group, to which Johannes Schemmel and also Sebastian Schmidt belongs. And I finished my PhD only very recently, and it was mostly about closed loop experiments on the BrainScaleS-2 platform. So I will start with the brief notes about BrainScaleS-2. So when we talk about this, we basically refer to these 65 nanometer chips. Johannes already said a lot about them, but here you can see two pictures of two different chips of the family. So on the left side is the smaller prototype version that contains 32 neurons with 32 synapses each. And on the right side, you see the full-scale version with 512 neurons. So the special thing about BrainScaleS-2 is that it has a hybrid architecture. That means it's not only the analog or mixed signal neural network core, but it also contains a digital CPU that can implement all kinds of code that you can write for it. So this scheme of operation is illustrated here. You have on the one hand this analog neural network core with these highly configurable 8x neurons and 6-bit synapses and all kinds of other stuff. And on the other hand, you have this CPU that runs with up to 400 megahertz clock speed. It has some on-chip RAM, which can be extended. And it also has a special vector unit extension that offers parallel access to for example, the synaptic weights on chip, but also to some correlation measurements that can be conducted with these synapses. So this architecture basically constrains the architecture of the experiment that you can implement, or the architecture of the code that you implement on your chip, and that's illustrated here. So for your algorithms, this has consequences, of course. You want to run your neural network, of course, on the neural network core. Because you don't want to do the number crunching on a digital CPU, then you can as well use a computer. But the digital CPU is very useful for implementing slower tasks, like, for example, learning or homeostasis rules, but also, for example, virtual agents like virtual robots or virtual embodiments. So the strength of the system is that you have this very versatile CPU that can implement all kinds of stuff, and you have this very fast and streamlined analog neural network core that can do the number crunching for you. So this is the special thing about the BrainScaleS-2 architecture. And another special thing that Sebastian mentioned in the talk before already is the speed up factor. So BrainScaleS-1 had a speed up factor of 10,000. BrainScaleS-2 has a speed up factor of only 1,000. But I think it's worth like talking a little bit about that. So on the one hand, this acceleration factor, of course, presents a huge advantage, because you can compress 10 years, for example, into a timescale of only 3.5 days. And that basically unlocks a whole field of research, because suddenly you can study phenomena like developmental phenomena in brains in a batch-like fashion, because you can execute them within days or hours instead of years. So this is great because it greatly improves your experiment throughput. But on the other hand, it of course poses a great challenge, because the smaller timescales, they become even smaller and harder to handle. So on the right, this is just a simple illustration. So here you can see a fruit fly that is confronted with a fly swatter at t = 0. And then you can see that it takes approximately 200 milliseconds for it to initiate motor reactions. And then these motor reactions, they take another like 80 milliseconds or so. And then one wing flap is on the order of a millisecond or 2 milliseconds. So if you compress that to the BrainScaleS-2 time frame, this whole procedure would not occur 3 times per second, but 3,000 times a second. And one of these flies would take off 3 times while this fly flaps its wings once. So that means if you want to keep the latency low, like, for example, 2 milliseconds is a latency that occurs in the brain, you have to compress it down to 2 microseconds. So these are basically the two most special things about the BrainScaleS-2 system when it comes to closed loop experiments. So now I want to present to you 3 experiments that we conducted on this platform. And the first one is Neuromorphic Pong. So this has been implemented by Timo Wunderlich, who I guess is also currently listening to this talk. And he's an expert with this because he did it. So in case you have questions regarding that, ask him. The experiment works like this. It's not actually a 2-player Pong, but a 1-player Pong, or Pong squash, as you might call it. And you have this ball that flies over a virtual field, and you have a receptive field. So basically, one presynaptic partner would be responsible for one of these rows, and then you have a connectivity matrix to the output neurons that steer the pedal. So if this neuron is most active, the pedal gets steered to this position. Now, the PPU does not only implement this game of Pong and the translation of spikes into the steering commands, but it also implements a learning rule that lets the network start with a random connectivity matrix in the first place, and then end up with a connectivity matrix that can successfully play Pong. And this is illustrated on this slide here. So here you can see a video. I hope it's a little bit fluent. So you can see the playing field on the left and the connectivity matrix on the right. What we would expect is a diagonal matrix after successful learning. And right now, this simulation is slowed down by a factor of 100, just to make it visible. But now the slowdown factor is decreased to only 2. So this is half the time that the system would be capable of. And you can see that 1,000 games of Pong are played approximately within 1 second. So the experiment throughput is very high. And after a few seconds, you can see that the system has already learned playing Pong. Yes, I cannot go any deeper because there are 2 more experiments that I want to show you. But if you have questions, please come back at us later. So the next experiment is neuromorphic insect navigation. And to understand what I mean by that, it's necessary to tell you a little bit about insects first. So on the right-hand side here, you see the desert ant Cataglyphis fortis, which is a pretty remarkable organism because it's able to navigate in the deserts where you don't have a lot of visual clues around. So here you see a typical trajectory. It leaves its nest. It can travel like a few hundred meters until it finds food. And then it returns to the nest, but it doesn't return on its pheromone trace or something like that, but it has something like a vectorial representation of the location of the nest. And it really goes straight back there. And now you can perform a lot of interesting, sometimes silly experiments to find out how these ants are doing that. And one mechanism that they use is called path integration. In nautics, you also call it dead reckoning. And this works by knowing your current heading direction and your current speed locally. And from like if you have this information all the time, you can take samples and then you can add it up or integrate it up, and then you can reconstruct your current position with respect to the starting point. So what you require for this is compass and an odometer, which is nothing but a way to measure the distance. So the past 10 years have produced a lot of scientific insights into insect brains, mostly by means of calcium imaging. Some of the stuff was known before, but like amazing details turned out recently. And what is known so far is that insects use, for example, the position of the sun to infer their current heading direction in space. Some insects, like the dung beetle Scarabeus, they even use the Milky Way at night to infer the heading direction. And all this information comes together in a very central part of the insect brain called the central complex, unsurprisingly. And this central complex actually carries a representation of a compass. So this is illustrated in the video on the right-hand side. What you see here is, again, a little fruit fly that is walking on an air-mounted ball. And on top of the fruit fly, you see a real-time imaging of its brain. So the bright parts correspond to neural activity in this brain region, and this is this protocerebral bridge over here. So this has like two compasses that are mirrored. And the blue bar around basically shows the actual heading direction of the fly. And you can see that this neural activity follows the current orientation of the fly. So this really works, and this is a structure that is very preserved among all kinds of insects or other arthropods as well, also crabs and lobsters and spiders and stuff like that have it. So this compass information is really present in insect brains. And the second component that we require for path integration is, of course, the speed indicator. And you can perform similar interesting experiments and also find that. So in this experiment conducted by Stone and others, the authors took a nocturnal tropical bee and put it into an arena where optical signals could be projected around it. And then they used normal patch clamp measurements to get the spiking activity of some neurons. And they found that some of these neurons are really sensitive to the optical flow across the eyes, and in a way that is really proportional to the field. And then there is another class of neuron that does the exact opposite, which basically accounts for the opposite direction. So both the compass information and the speed indicator is present in the insect brain, and you can even take that one step further, do more measurements, and come up with an entire neural model of how this path integration scheme is carried out in the bee brain. And this is what the authors of the same paper have done. So down here you see the trajectory of a virtual insect in this case with a simulated brain. And it performs a random walk, and after some time it is allowed to return home to its nest. And then you can also see nice emergent behavior, namely that it starts looping around as soon as it reaches this nest, like a fly in your room. Yes, and the original network was composed of only just below 100 neurons, something like that. But at the time, we had only the smaller prototype chip available with 32 neurons. So the first challenge of porting this experiment to the hardware was to reduce the number of neurons. And this actually can be done straightforwardly. So the original network had 8 directions, so a resolution of 45 degrees, basically. And this could be straightforwardly reduced to only 4 directions with a 90-degree resolution. And then we had some other benefits, like, for example, we didn't have to stick to Dale's law, and therefore we could just remove an entire population and make this population here both excitatory and inhibitory at the same time. So the part on the right, which is basically this bee, including all its sensors, is implemented on the digital coprocessor. And the right part here, which is the neural network, runs, of course, on the neuromorphic core. And the first layer here is implemented as mixed signal circuits. So spikes are injected from this CPU into the network, and the rest are really actual neurons. And these output motor neurons here, they provide propulsion to the left or right-hand side of this agent and steer it similar to a tank. Yes, and of course, I also have a video of that. So what you see here on the left-hand side is the trajectory of the insect, in gray the outbound journey, and in blue the return trajectory. It starts looping around. And on the right-hand side, you see the brain activity that is going on in parallel. And in this top left corner of the trajectory window, you can also see the time scales. And you see that this whole experiment takes 200 milliseconds. So when you multiply that by 1,000 to get the actual speed up factor, then you have 200 seconds, which is just above 3 minutes or so. So that would correspond to the entire insect journey, but you can compress it to just 1/5 of a second. Yes. And if you execute that in real time, it would look like that. So this is actually the throughput that we get on the hardware. The breaks in between the single runs correspond to the time that we need to get the data back from the chip to the host computer. Yes. Now, the third and final experiment that I want to show you is an actual robotic system. It is called the Playpen, or like in this case, the Playpen 2, because it's the second iteration. And it's a pantographic robot. That means the actual agent does not move on its own, but is driven by these two arms. And the agent is basically a set of sensors that moves over a surface. So this surface could contain a pattern, like, for example, a maze or a racetrack or a potential or whatever you can imagine. And these sensor signals, analog sensor signals again, are translated by a digital processor into spikes, which are then processed by the neural network again. So the system architecture looks like this. You have the BrainScale system over here, which is composed of the chip itself and an FPGA. This FPGA can send spikes out to the system, and it can receive spikes from the system. And then you need something like a pulse shaper that makes these spikes usable for a motor driver that steers these two arms into both directions. So each motor has two channels for each direction. And then the sensor information that is gathered from the arms and from the sensor can be returned, first amplified by 6 programmable.
Excuse me.
Sorry, yes.
We have about 3 or 4 minutes, including questions. So it probably should come to an end.
Oh, don't I have six? I thought I'm at 16 minutes right now.
You should have 3 minutes left, and then plus 5 minutes for questions.
Yes, that's fine. That's fine. Yes, so you have these 6 programmable gain amplifiers, and these can first translate the voltages, and they can be digitized by a microcontroller, which produces spikes again, which then get processed by the signal again. Here is another slide that just shows some details of the robotic part. So you have these little sensor that can measure the arm rotation. It's just a magnetic Hall sensor to save weight on these arms to keep them very fast. And on the bottom, you see this actual sensor head that you can think of as an agent with 3 photoreceptors here and some preamplifiers over here for enhanced signal fidelity. So one experiment that you can perform with this, perhaps the simplest experiment, is robotic gradient descent. So you translate these photo signals directly into spike rates and feed them into a tiny neural network with only 4 neurons. And each of these neurons is responsible for one direction. So what is happening in the end is that the sensor head is steered to the maximum of a light source below. And here you can see the trajectories in 2-dimensional space. It's 4 trajectories. They start in the 4 corners, and they all manage to hit the center of the light. And of course, I also brought you a video in this case, and I guess that's not very fluent on your screens. So it's moving very, very fast, and it's doing the same task repeatedly. Like one return to the center takes approximately 200 milliseconds again. These flashy LEDs, they indicate the spiking behavior. Here you can see the BrainScaleS-2 platform. And this experiment does not just run repeatedly in this case, but it is actually an evolution that is going on. So in this case, the network started with a random configuration, and we use an evolutionary strategy to make it find the maximum on its own. Yeah, and this you can see on the next slide. So the background of these figures over here indicates the light. So the bluer, the brighter. And here you can see the network performance, and these indicate the connectivity matrices, just so you see that something happens. And the black lines here indicate the final positions of the sensor head. So you can see that after 100 generations, actually after 20 generations already, it is able to reach that goal. So let me conclude with a short summary and outlook. On the one hand, there are 3 things that I find worth mentioning. One is that accelerated closed loop experiments are actually feasible. There's like, it's not so clear whether you can interface a setup that runs 1,000 times faster than biology with a robotic setup, because they are mostly made for smaller timescales. But in this case, it really works, like we demonstrated this. And if you go virtual, then there is no issue at all. I would even go further and claim that studying learning rules in cybernetic systems, that is in closed loop systems, actually requires acceleration. Because if you have an experiment throughput of a normal robotic system, which behaves like on a human-like timescale, then you can wait weeks for these systems to learn, if they learn at the same speed as a human learns, for example. And the last take-home message, so to say, is that 512 neurons are actually a lot. So this path integration network that you've seen works with 17 neurons. So 17 neurons is enough to perform two-dimensional path integration. And there is a whole world of experiments that can be conducted with an actual low number of neurons that easily fits on one chip. So we don't have to go to insanely large scales. This is, of course, necessary for some tasks, but not for all. There's plenty of room at the bottom, as Feynman might have said. Yeah, and the future plans is, of course, to have more insects, I hope. For example, this path integration scheme can be extended. There is concrete plans to implement a neuromorphic motor controller, which is not feasible with a real-time system, but requires this acceleration factor. And then you can, of course, go to more complex robotic tasks like steering quadrocopters and so on. Yes, and then I'd like to thank you for your attention, and I'm happy to receive your questions.